of plant growth and development. Okay, so I hope all of you might be aware. I think that what is the main role of this arbuscular mycorrhizae in nature, right? Okay, so to begin with, just I will discuss how exactly the arbuscular mycorrhizal research came into forefront of scientific research, particularly when it comes to plant growth and development. Actually, so it is believed that the first person to describe this arbuscular mycorrhiza is Megeli, the very same person who first coined the term meristems, then xylem, phloem. So all those, most of the scientific terms, he is the person who is responsible for developing and coining the concepts. And he is the person. It is believed that he is the one who first described this arbuscular mycorrhiza in 1842. During the same year when this Mendel and even Darwin were there during that period, actually, and followed by his brief description about this arbuscular mycorrhizae. Next, the people who worked on extensively worked on this particular mycorrhizae, and they have observed different structures within the plant body, and they found that these organisms might be pathogenic in nature in the beginning. Actually, the people who initially worked with regard to this mycorrhiza were with anatomical descriptions, and also most of the information was given by Slidge in 1889, and followed by Dangier, JNC, then Terry, Petrie, then Gallard, then Perunel, Jones, and Lehman. It is believed that the information is available with regard to the structures, that is anatomical relationship, was first proposed by this basic concept of arbuscular mycorrhizal and association, that these are the fungi, was first given by Slidge in the year 1889. And again, Jense was considered to be the first person to observe the vesicular-like structures within the body, which you can see within when you stain this mycorrhizae with trypan blue or various other types of stains, and which is followed by observation of arbuscules by Gallard in the year 1905 in the inner cortical region. And it is Gallard who made accurate observations with regard to this arbuscular mycorrhizae, and he came to certain conclusions that the host, the membrane, that is plasma membrane, is completely surrounded by these arbuscules, which can be observed within the cells of any higher plants of interest if people are working, actually. And it was Cox and Sender who confirmed this observation by using transmission electron microscopes. Okay, in the year 1974, I think the concept was proposed by Gallard in 1905, but this was later confirmed by the Cox and Sanders in the year 1974, and they proved that they concluded that yes, these arbuscules are surrounded by membrane of host. And the term mycorrhizae was coined by Frank in the year 1885 itself, and he used a single R actually when he initially proposed that word, and later on another one R was added by Kelly in the year 1931 actually. Okay. And again, Frank in 1887 recognized two different types of mycorrhizas when he encountered two different structures actually when in plant roots. One he named as one is ectotrophic in nature, and another one is endotrophic in nature. And he was mostly concerned working on ericaceous and orchid mycorrhizae. And when you stain any root, actually, for that matter, even almost if you talk about mycorrhizal association, in 90% of the plants, actually, earlier they were thought of 80%. Now 90% of the angiospermic plants or the flowering plants have the association with this vesicle arbuscular mycorrhizae. These are the structures one can observe, actually. These within the cell, the arbuscules will be present, and vesicles will be there either intracellular or intercellular in the nature. And these vesicles are considered to be reserve food materials. They store reserve food material during this desiccation times, you can say. And the name vesicular and arbuscular was initially because of these structures, actually, vesicles and arbuscules. The name was given as vesicular and arbuscular mycorrhizal fungi. Of mycorrhizal fungi associated with these plants. This name was there until very recently, I think, even till 1990s. And because most of the people thought that both vesicles and arbuscules are present within the mycorrhizae. But of late, people recognize that vesicles, not all mycorrhizal association are forming these vesicles. And it was proposed that the name vesicles must be deleted or removed, and only arbuscular mycorrhizae can be retained, actually, to name this fungi, group of fungi. And in the year 1859, actually, in the for the beneficial aspects, when it comes to beneficial aspects of this mycorrhizae, Baylis was the person who first reported that this mycorrhizae is the main media or main what you can say, this the mycorrhizal association formed by these plants is mainly for the uptake of phosphate, actually. So he demonstrated this particular concept. He proved that the mycorrhizae mostly they help in uptake of increase uptake of phosphorus. I think till then people were under the impression that this mycorrhizae is mostly, I think, in the beginning, actually, they were thought that this nitrogen uptake, they help in nitrogen uptake. It was Baylis in the year 1959, he disproved, and he proved that not much nitrogen, but phosphorus uptake is helpful for this mycorrhizal association to the plant, actually. Okay. When it comes to this when we talk about evolutionary significance, or when exactly this mycorrhizae started forming fungal association with the plants, actually. So to this question, the answers one can say is once the plants started moving from this aquatic mode of life to this terrestrial mode of life. So they started facing a lot of problem, particularly with regard to acquisition of nutrient or soil moisture, and also to acclimatize themselves to this dry condition. They were facing even desiccation problem. So all these problems led to this symbiotic association with this plant. When we screen the terrestrial plants, actually, most of the terrestrial plants as of now, right from this bryophytes, pteridophytes, gymnosperms, and angiosperms, if you talk, when all of them, they have this mycorrhizal association. And that is mainly for the survival of that particular plants. This association might have developed. This was the concept put forth by most of the scientists who are working. The hypothesis says that these terrestrial plants are the products of ancient and continuous symbiosis of semi-aquatic ancestral green alga and this aquatic fungi, actually. So even when there was a transition from moving the plants moving from this aquatic life to this terrestrial mode of life, I think I think since then the association started. And initially this Mycorrhiza fungus, I think even now the Mycorrhiza fungus I think they are the obligate nature is considered to be that association actually. Okay? And it is believed that this Silurian and Devonian explosion of colonization of plants, particularly land plants, that is considered to be the era where this evolution of land plants took place, and that was considered to be perhaps that particular era is also considered to be the era of mutualistic partnership between the terrestrial plants and these fungi, and particularly to cope with the problems associated with the desiccation and starvation and its own survival of the plant actually. Okay, and in addition that the transition of plants from this predominantly aquatic to terrestrial lifestyle was considered to be the most historic event, and greening of plant, what we can say, greening of globe. This was as of now, there is a concept that this greening of globe wouldn't have possible until I think unless the fungi formed an association with this terrestrial plants actually. So, okay, so in other words, so we can say that the greening of globe has taken place because of the association formed between the different groups of plants actually. And as you know, these lichens, they are the symbiotic association between plant, this fungi as well as algae. So in any this succession, if you talk about succession, any succession begins with this lichens actually. Okay, so that is the main thing I think one can discuss here actually. The association between the fungi as well as plant, and this table, this chart shows that you can see here early in the Devonian period, you can see 419 billion years, million years ago, these AM-like structures were observed in some of the plants of earliest plants reported on this earth. Almost this association dates back 419 million years ago actually. Okay, so you can see here this is a star. So from then onwards, these different organism plants took different deviations, right from bryophytes, then moved to the change over to pteridophytic nature, then gymnosperms, then angiosperms. Presently, most of the plants, what we can see here, angiosperms, and the association is almost 420 million years ago. That is the mycorrhizal fungi as well as plants, you can see. Okay. So when we talk about the association of different microorganisms with plants, this AMF is considered to be the most ancient one. So you can see this is almost 400 between 500 to 400 million years ago. You can see this mycorrhizal association was dated back. And when it comes to this is endomycorrhiza. What we are seeing, the oldest one among the mycorrhiza. There are two different types of mycorrhizas, ectomycorrhiza and endomycorrhiza. Among these two, endomycorrhiza are considered to be the oldest one when compared to ectomycorrhiza. Ectomycorrhiza, they formed between 300 to 150 million years ago. And this rhizobium and frankia, they are very recent actually. You can see rhizobium dated back till 100 years old and 100 million years old, and this frankia, after this rhizobium formed, frankia were dated back actually. So this is to this chart. Just I wanted to say that arbuscular mycorrhizal fungi are considered to be the oldest association when compared to the symbiotic association of different organisms with higher plants. Okay. And just you can see for the transition of plants from the aquatic life to this terrestrial mode of life. So for the terrestrial mode of life, the plant has to acquire nutrients from the soil. So that is the main important thing because the mycorrhizae are known to have produce certain compounds which can help them to, or you can say the mycorrhizae sends the mycelium to different areas actually. Both ectomycorrhizae as well as endomycorrhizae, they expand the root horizon so that the plant roots may not reach to that distance and acquire nutrients and moisture. But mycorrhizae played a very important role in establishing plants, you can see here. So the aerial growth of the plant can be attributed to the mycorrhizal association. That is the main thing one can draw from this particular figure. So the lifestyle shift, as I mentioned, lifestyle shift from aquatic to this terrestrial mode brought with number of challenges related to this nutrient acquisition, then water acquisition, as well as volatile environmental changes. So that change to overcome all these challenges, the earliest plants like bryophytes entered into mutualistic symbiosis with that of arbuscular mycorrhizae. And since then, these organisms are helping the plants to in different ways actually. So normally, as of now, I think most of the experiments conducted with these mycorrhizal associations, they have proved that the mycorrhiza plays a very important role in phosphorus acquisition. That is the main function. In addition to that, the mycorrhiza also plays a very important role in overcoming various what you can say hazardous, harsh environmental effect on the plant, as well as overcome diseases through this competitive mode of for resources. Then it also proved that the mycorrhizal plants are known to acquire more nitrogen when compared to non-mycorrhizal plants. So all these are the benefits the plants can get if they form symbiotic association with plants. And so the structures what we observe within the plants, the mycorrhizal associated plants are different types actually. So either you can see the only mycelium within the plant if you are dealing with endomycorrhizae, and then you can see vesicles and the arbuscules within the plant structures. And even in the rhizospheric soil, you can find spores actually, the spores which are produced by the mycorrhiza. So these are the structures one can observe. And for categorization, the mycorrhizas have been differentiated into different categories actually. So they are ectomycorrhizae, classification aspects when it comes to ectomycorrhizae, then arbuscular mycorrhizae, then orchidaceous mycorrhizae, and ericoid, then arbutoid and monotropoids. So all these are different mycorrhizae, and ectomycorrhizae, it is only restricted to epidermal cells or very few cells within the root surface and root and the ectom. Why it is called ecto means it will not send any the mycelium or hostorial like structure within the cell, so that is why they are called as ectomycorrhizae. Endomycorrhizae, it sends these arbuscules, hostorial like structures for the exchange of food material. That's why they are called as endomycorrhizae. Okay. So just to differentiate between this ectomycorrhizae, the above one is the angiospermic ectomycorrhizal association, and the below one you can see this is association of with conifer plants. So you can see in ectomycorrhizal association in angiosperms, it is restricted to only epidermal region of the root, and it forms a net around this root. The mycelial network actually forms around the root. In conifers, though the mycelium enters within the few cells of cortical region, but it will not send its mycelium or the hostorial structure within the cells actually. So it will be in between intercellular spaces. So that is why they are called as ectomycorrhizae. Okay? So this is LS and this is TS, you can see here. So in angiosperms, it is restricted only to epidermal cells, you can see here. And it forms a heavy network, this mycelial network, just exterior to the root. And in conifers, the mycelium is in between the cells, intercellular spaces. It will occupy the space, intercellular spaces in the cortical region. So another one type is ectendomycorrhizae. So which shows both characteristic features of endomycorrhizae as well as ectomycorrhizae. You can see some of the mycelium are entering within the cells, and most of the mycelium is occupied intercellular in space nature. So these kind of structures are can be observed, and these organisms are called ectendomycorrhizae. So you can see this ectendomycorrhizae is covering the entire space, as well as part of the mycelium is entering within the cells actually. So you can see this mycelium gets within the cell, and it forms a this Hartic net type, what you can say, net-like structures of arbuscules. And these arbuscules displace this. It is not damaged. It will not damage this protoplast of the host plant. It will just push around, and the surrounding this area, the protoplast will be occupied. And this is the place where the exchange of food material takes place. And it forms intercellular space. The mycelium network is occupied between the intercellular space, forming a Hartic net-like structure actually. So this is considered as a Hartic net-like structure. Okay. So these are the structures one can see. This is the mycelial network in the cortical region, and it runs parallel to the cells, and the mycelial network will be intercellular in nature. This AMF, and it produces spores and arbuscules and vesicles. So both you can see here. So within the mycorrhizal association, based on the structure of arbuscules, they have differentiated into two types. One is RM type and another one is Paris type. So this in RM type of arbuscular mycorrhizae, you can see these arbuscules are tree-like projections actually. You can see the structure is varied, slightly different. So in the beginning, it will be this Paris type, but ultimately it will form a tree-like structures within the root actually. So these structures, if you are observing, then it is called as ARM type of arbuscular mycorrhizae. And in Paris type, it forms a coil structure, and over that coil structure, it forms a this finger-like projections you can see here. So these structures, if you are observing, then you can categorize them as Paris type arbuscular mycorrhizae. Okay. So vesicles will be intracellular in nature in case of our Paris type. Here also the vesicles will be intracellular or intercellular, you can say. So coming to this ericoid mycorrhizae, so most of the ericoid plants, they form this endomycorrhizal association, and you can see this is the network. The whole of the cell is filled with this the arbuscules structures. It is a hyphal complex it has formed. So this hyphal complex is the place where the exchange of food material takes place. So once this mycorrhiza dies, I think this hyphal complex will be digested, and additional nutrients will be supplied to the host plant actually. And this is with regard to this arbutoid mycorrhizae. So in addition to that, you will also find this Hartic net type structures actually. So you can see here in between the intercellular spaces. So whole of the area is covered with the mycelial network of mycorrhizae. Coming to this monotropoid nature, in addition to this Hartic net-like structure, it produces a peg-like structure, you can see. The fungal peg-like structure for sending. So instead of this coil structure, it produces a peg-like structure, And this is the area where the exchange of food material takes place. These are the characteristic features of monotropoid mycorrhiza, and these are because it sends the mycelium within the cell. They are also grouped under this endomycorrhizae. And this is orchid mycorrhizae. As you all know, this orchid is known to produce the seeds which are recalcitrant in nature. So as soon as they are shed from the leaf, because they lack endosperm, they have to germinate. So soon after their germination, if they form, until and unless they form association with mycorrhizae, they have demonstrated that there are so many reports they have been demonstrated that the orchid seedling will not survive at all. So the orchids seedlings which form association with mycorrhizae are known to survive beginning till the orchid plant is established itself and starts synthesizing its food. So I think still the establishment of this initial stage, mycorrhizae plays a very important role in supplying nutrients to the plant. So once the plant becomes self-sufficient for synthesizing its own food, the mycorrhizal network or the mycelial network will be digested within the host, and it is utilized, and the additional nutrition will be acquired. Actually, this is with regard to orchid plants. Okay. So coming to this, the importance of arbuscular mycorrhizae, the most of the mycorrhizal association, particularly arbuscular mycorrhizal, as of now, people are very much interested, because most of the input we are adding, actually they are causing a lot of problem, and these mycorrhiza are considered to be the safest organism, because they have been in association with these terrestrial plants since so many years. So most of the strain specific and high this efficient strains are they are looking for, particularly for this helping the plants in acquiring this phosphorus, copper, and zinc. Actually, zinc. So all these elements are immobile elements, and these immobile elements, until and unless these are present within the vicinity of the roots, the plant cannot absorb from the distance, and they are not they are not immobile in nature. That's why the mycorrhizal the mycelium, which is forming association with plants, it can reach to these particular the areas where phosphorus and zincs are available, and it solubilizes, and it greatly benefits the plants for assimilating these particular nutrients to the plants, actually. So that is why mycorrhiza are considered to be very important at global scale, you can say. So as I mentioned, 80 to 90% of all extant plant species are have this the AMF, the arbuscular mycorrhizal mutualistic symbiotic association, and this particular association is thought to be approximately 480 million years ago, and that is once the plant started moving from the aquatic mode of life to this terrestrial mode of life, then only the association started. And the EMF are considered to be obligate biotrophs that fall within this glomeromycotina of subphylum Mycorrhiza, actually. So because they have a unique features, these mycorrhizas are considered to be, as of now, they have separated a separate phylum called glomeromycotina. Okay? And this glomeromycota represents almost 1,000 species, and out of which only approximately 300 species have been described based on this morphology, so morphological basis. And it is relatively a small group of fungi which forms this obligate symbiosis, and with almost, you can see here, As of now, the 2 lakh species globally described species, it forms very low because of its low host specificity actually. And AMF symbiosis appear to have emerged at the same time when these biophytes, particularly mosses, liver dots, and horn rods, diverged from a single lineage of single-celled algal chlorophyte approximately 440 to 530 million years ago. And some of the earliest fossil records, they also confirm that this association is approximately, they have reported actually the plants, they have from this Rainy chart or Rainia like plants, they have reported almost from as old as approximately 407 million years ago. They have shown, and the symbiosis is formed when the fungus penetrates the plant cell wall without damaging this plasma membrane and forming intricate branches like arbuscules within that structures. So you can see here, this is the plant Oniophyton lignaria, and the arrowheads, what you can see here, these are the structures where arbuscules like structures they have observed when they took the anatomical sections of this particular plant. So this plant is almost 407 million year old plant. They have recovered from Rainy charts actually. Okay. And this is the another one structures Mycorrhiza of this mycotian type of corm actually. You can see here, this is the corm. And you can see some of the structures inside this. The arrowhead shows this mycorrhizas. Okay. So all these findings indicates that this association is almost more than 400 million years old actually. So I think these are some of the pictures you can see. They have taken from the plants. They have collected from Rainy chart plant fossils, and they are considered to be as old as 400 and more than 470 million years old. And what all these why I'm showing is these mycorrhizae played a very important role, crucial role when the plant started moving from aquatic mode of life to this terrestrial mode of life. And this association has co-evolved along with the plant actually. That's why they are some people argue that these are pathogenic in nature, but they are not pathogenic in nature. They are the symbiotic, so they have the symbiotic association actually. So coming to this application aspects, why we need to study this mycorrhizae, what are the important aspects? So you can see here the mycorrhizae has got application in different areas. You know, you can study the mycorrhizae plays a very important role in what you can say, can the mycorrhizal plant show pathogenic resistance? Then it enhances water uptake, then microbial, it also increase the microbial diversity, then soil conservation aspect and nutrient availability. And that also accumulates the active ingredients, then gives stress tolerance to the plant, then helps in nutrient uptake, then disease resistance and crop productivity. In addition to that, they also mitigate climate change. Then they can help in phyto remediation, then reclamation of degraded land, and this ecosystem biodiversity. That is, they help in designing, or you can say developing a particular ecosystem. That is also one of the important aspects. So that's why mycorrhizae plays a very important role. So in almost all approaches, mycorrhizal association are considered to be very good organisms, and these are very important organisms one has to study actually, look for actually. Okay. And mechanism of plant root colonization and arbuscular mycorrhizae. How exactly within the root the plant appears or looks at that. If you want to see the diameter of the mycorrhizal mycelium is relatively very small when it comes to physical appearance, and then the plant root or head, the root hairs, and hence it is considered to be more explore soil and soil aggregate much efficiently when compared to plant root alone actually. Okay. And how exactly it forms association is the germinating spore. They physically come in contact with the root surface of the plant by forming a structure called hypopodium. And from this hypopodium, the hyphae penetrates to the intercellular space, or the intercellular space, and it enters into the cortical region, and it forms the mycorrhizal network within the cortical region, and then the endomycorrhizae enters within the cell, forming arbuscules, and the exchange of food material takes place. So you can see here the spore is germinating, and this germinating forms comes in contact with this hypopodium, and because of the calcium spiking, the signal transduces to the interior of the cell, and the plant recognizes that this particular fungi which is coming in contact with the mycorrhizal, the root surface, is symbiotic in nature, and it allows the entry of this pathogen, this mycelium, to gain within the cortical region. And once it reaches the cortical region, it sends this part of this mycelium within the cells, and the mycelial network forms, and here is the place. You can see this is the place where the exchange of food material takes place. Okay. So again, this is a different view. You can see here the differences between this ectomycorrhizae and endomycorrhizae. Just you can, if you want to see, the ectomycorrhizae is it will not send any sort of mycelial the mycelia within the cell actually. So it will occupy a intercellular space, or it will, most of the plants in angiospermics, it will be the mycelial network will be only on the restricted to epidermal cells, and it forms a mat-like structure above the roots. This is ectomycorrhizae. Whereas endomycorrhizae is it enters within the cortical region and it sends a mycelium. So that is the major difference. So you can see here the exchange of food material. Just I want to show with this diagram that how the partners get benefited actually. The exchange of food material takes place. The mycorrhizae supplies phosphorus to the plant, and in turn it gets carbon source from the host actually. So here you can see here this peri-arbuscular membrane. In this peri-arbuscular membrane, all the activity is known to taking place with regard to either exchange of food material. You can see here. Okay. So coming to this biochemical aspects, how the signal transduction and how the plant recognizes in that vicinity of the root whether the organism or the spores which are present, whether they are beneficial or harmful, the plant recognizes. That is based on the biochemical signals exchange between the plant as well as the mycorrhizae. So the same the exchange of signal is similar to that of the rhizobium legume symbiosis exchange. Here in case of mycorrhizae, the plant releases certain secondary metabolites, and in response to that, the germination of spores takes place. So the root exudates that is released from the plant. So the resting spores will be germinated, and the mycelium will move towards that particular direction actually. So that is how the signal transduction takes place, and it is believed that this strigolactone is one of the signaling hormone which plays a very important role in the exchange of these signals actually, because strigolactones are considered to be one of the important signals, which is one of the this phytohormone recorded out of the, I think, 9 or 10 phytohormones. This strigolactone is one of the important phytohormone. It sends the signal, and in response to that, the spores germinates and it moves towards the root area and forms association. So that is how, just I wanted to see depict this with this picture. So as I mentioned, the attachment of pre-symbiotic mycelium is mediated through this host root exudates, particularly strigolactone, and in response to that, the spore germinates, and it comes and forms hypopodium on the surface of the root, and from where the pre-penetration apparatus appears, and it enters into the cytoplasm of the epidermal cell, and again cortical cells actually. So in the intracellular hyphae, they branch profusely and forms a structure called as orbuscules, and these are the structures which plays a very important role in exchange of food material. So same thing you can see here. So in response to strigolactone, germinates, attachment, and from there it enters to the epidermal cell, and from there it enters again the cortical cells, and this within the cortical cell it forms a orbuscules. So when it comes to this molecular mechanism of this mycorrhizal symbiosis, 7 genes have been reported so far to be responsible for this symbiosis with the plants, and particularly the plant protein that code for receptor-like kinases, that is LJSYMRK, and which is reported first from Lotus japonica, and MTDMI2 in Medicago tranquillata plays an instrumental role in directly or indirectly recognizing both rhizobial as well as mycorrhizal signals. And through this perception of this receptor proteins, the signal perception, the signal transduction takes place, and through this phosphorylation of kinase domain, and within the cytoplasm of the plant, and that will in turn help in forming this mycorrhizal association. Normally, these mycorrhizal symbiotic signals are the mixture of effector proteins and chitin derived compounds such as chitin oligosaccharides and lipochitinosaccharides actually, and plant receptors of this LCOs are which activates this calcium oscillation in the epidermal cells and within the cells, and within that the plant nucleus, through this well conserved set of genes, the part of which are referred to as common symbiosis signaling pathway, will be triggered actually. So as soon as this pathway is triggered, the symbiosis is initiated, and AMF are known to produce a distinct biologically active compound, which in turn induce different pathways, and AMF known to release these MYC factors, which are similar to that of NOD factors of rhizobium, and during the symbiosis, and they are considered to be responsible for host root colonization in the presence of strigolactone. So this MY factor in response to this strigolactone is known to form a symbiotic association. So again, the same thing, I think this we can skip. So you can see here this mycorrhizal LCOs in response to this sligodactylon. It germinates and it forms a epipodium and sends the mycelium to this interior of the plant. So this is with regard to the gene expressions. So once the mycorrhizae comes in contact with the any cells, these are different genes which are expressed actually. In the stage 1, PPA is expressed, then stage 2, it gains a cell entry, and stage 3, it forms a bird footstep structures within the host cell, and in stage 4, you can see this mature arbuscules. And in stage 5, the arbuscule is collapsing, and once it collapses, it forms a septum here, and the whole of the collapsed mycelium will be digested by the host plant, and this additional nutrients will be again the host cell will get additional nutrients in addition to whatever the phosphorus is being fixed when the mycorrhiza is in active form actually. So this is the added factor you can see here. So in order to distinguish the AMF and the complex signaling exchange pathways, and these signaling pathways are very much essential because in addition to AMF, there are so many pathogenic microorganisms which are there within the this rhizospheric zone. So genetic evidence has suggested that many of the signaling compounds of plants were present even in advanced chlorophyllene algae, some of which are they are terrestrial in nature actually. So coming to this role of mycorrhizae in sustainable nutrient mobilization and crop nutrition, that is the application aspects. So this role of the extended hyphal network is considered to crucial in uptake of nutrients because plants are known to produce roots or root hairs only to certain extent. So in order to get the accessibility, the plants forms association with mycorrhizae, and it gets a wider rhizospheric area for acquiring nutrients actually. So the length of they can be extended for several centimeters in soil, and the total surface area can be manifold greater than that of the host root. So that is the main criteria for the getting larger nutrients when compared to non-mycorrhizal plant. And a larger surface area allows the plant to better acquire phosphates and other minerals, nutrients from the rhizospheric soil. So you can see here. So if this is the root hairs might be several centimeters or several centimeters in length actually. But the mycorrhizal network, you can see it can send to different more than I think manifold, the 10 times, you can say 10 times fold. And there are reports that I think I read in one of the articles that ectomycorrhizae, it can send its mycelium to almost 100 meters or more than 100 meters also. So that is the area where it covers and acquires nutrients, and in turn it supplies to the host, and it gets nutrients from the host. That is the association you can see here. So the uptake of nutrients, as I mentioned, the main place, the nutrient uptake, the exchange of nutrient takes place at this arbuscules, a place of arbuscule where periarbuscular space is there. That is the place where the exchange of food material takes place. So this is in case of the uptake of plants in the absence of mycorrhizae. So in the presence of this mycorrhizal uptake, you can extend the area. You can see the area where the length of this one actually. Okay. And the morphological characteristic features, as I mentioned, this is extra radical mycelium, and this is intra-radical mycelium. So you can see mainly what I want to say is the mycorrhizal association the extends the area required for exposing the plant to beyond the root area, actually root surface area, and it acquires the nutrients from different places, actually beyond the root zone area, you can say. So the mobilization of available nitrogen from the litter of the plant is also facilitated by this AM extra radical mycelium, and AM proves to be stronger candidate for low input sustainable agriculture, and through this active network of mycelium can help mobilize nutrients from the nutrient crop residue itself, actually. Okay. So these are just you can see the mycorrhizal association can help the plant to get nitrogen as well as phosphorus. Phosphorus transporter through this phosphorus transporter, then this ammonia transporter, then nitrate transporter. Even some of the amino acids can be acquired from the surrounding areas. Then urea. So in turn, the plant will get this carbon source from the host, actually. Okay. This you can see glucose, sucrose, fructose. It is taken by the plant from the plant. So in addition to that, mycorrhiza also plays a very important role in ameliorating plant health through this improved resistance of various abiotic and biotic stresses. Particularly the salt affected area soils, almost as of now this soil alkalinity is considered to be the major problem as of now. Most of the irrigated area, because and as of now I think 10% of the global land surface is considered to be alkaline in nature. The soil salinity has been increased. So to overcome this particular soil alkalinity, mycorrhiza played a very important role. Particularly, they act as the phytotoxic effect of this salinity, which includes lower water potential in soil solution, which leads to this decreased water uptake and ion toxicity related to increased uptake of this the chlorine and the sodium, and this reduction of mineral uptake and absorption. So plants have better sustenance against adverse effect of the soil salinity when colonization of when they form the mycorrhizal association. So all these can be overcome to this one. And the question is whether the AMF improves the plant growth under extreme adverse conditions. Definitely the answer will be yes, because the release of metabolites such as phytohormones, amino acids, vitamins, minerals, and solubilization processes, then providing nutritional and structural benefits to the plant. The mycorrhiza helps, and the production and accumulation of secondary metabolites, then osmotic adjustment under osmotic stress, then improved nitrogen fixation, then enhanced photosynthetic rate, and increase resistance against abiotic and biotic stress. Through this mycorrhiza helps the plant to overcome all these problems. And mycorrhizal fungi can improve this plant tolerance to heavy metals, drought, and salinity, and also protect plants from pathogens. So all these are the benefits the mycorrhiza can give once the plant forms association with this mycorrhiza. So a role of this AMF symbiosis in water stress, you can see to overcome water stress, mycorrhiza plays a very important role in the plant because they have this the physiological revamp process, then stress-related gene expression, the increased stress-related gene expression, it helps. Then hormonal balance through this oxidative burst control, then osmotic adjustment. So all these are some of the important aspects when the plant gets benefits from the mycorrhizal association. And this coming to this mycorrhiza colonized plant act as a drought tolerant as well as drought avoider. Actually, drought avoider, as most of the plants are able to adapt in moisture deficient environment through these two major strategies. Normally, this drought avoidance and drought tolerance, and proline is considered to be one of the important this osmolyte which is accumulated, and this the level of which is raised in different abiotic stress during this drought and salinity and cold stress actually. So when the mycorrhizal associated plant as well as the non-mycorrhizal plant, when the proline level were assessed, the mycorrhizal plants were considered to be accumulating more proline when compared to non-mycorrhizal plants. So that is how the plant helps in the mycorrhiza helps in overcoming this stress tolerance. So in addition to that, this superoxide dismutase, accumulation of superoxide dismutase, which is one of the most efficient antioxidant enzyme, which initiate detoxification of free radicals. So this level is also known to increase, and the increased level of peroxidase is also considered to be one of the important parameters to overcome the stress, particularly. They are found in chloroplast of mesophyll cells, which is known to carry out detoxification of H2O2 by ascorbate glutathione cycle actually. Okay, the hydrogen peroxide and glycol peroxidase, that is GPX, are a diverse group of enzymes which detoxify H2O2 in cytosol, and the mycorrhizal plants are known to increase the concentration of this also. Then improvement in soil moisture holding capacity is one of the important aspect. The mycorrhiza are known to produce a compound or a glycoprotein which is called as glomalin. So this glomalin is known to form this soil, what you can say, clumping or soil, the moisture holding capacity will be increased wherever this glomalin is more actually. So that has been demonstrated beyond doubt. So this AM fungal hyphae can enhance soil architecture due to this their extensive filamentous network. That is hyphal filamentous network, and in addition to that, they also known to produce glomalin, which helps in retaining soil moisture. Thus, this mycorrhiza is considered to be the best biological tool for improving soil structure. So, plant hormone regulation of AMF during this drought. Normally, the hormones which are regulated, this is a positive influence on the level of jasmonate, terpenoids, then carotenoids and phenols, which are known to increase differentiated enzymes like this phenylalanine ammonialase, and then polyphenyl oxidase and peroxidase. Okay, and these are that is AMF, the mycorrhizal plant and non-mycorrhizal plant. The concentration of these plant hormones are known to vary actually. So that is how mycorrhizal plants are considered to be more efficient in synthesizing these differentiated enzymes when compared to non-mycorrhizal plants. So there is a significance progress has been made in understanding the role of mycorrhizal symbiosis in elevating drought tolerance, but there are still missing links for fully decoding this mechanism, and the people who are interested in mycorrhizal research can look in this direction actually. Okay, there is a lacuna in understanding this particular mechanism, and whether this mycorrhizal symbiosis is helps in sustainable ecosystem services. definitely the plant, the mycorrhiza plays a very important role in sustaining the sustainable ecosystem services, particularly for the well-being of human beings, providing various resources and processes. Like trade-offs and links between plants and microbial communities can act as a diverse drivers for a wide range of processes of ecosystem, particularly, as I mentioned, during this succession, once the plant start this establishing in the drier areas, so mycorrhiza, the plants which form mycorrhizal association will survive and grow better when compared to non-mycorrhizal plants, and in turn, the diversity of the plants can be increased actually there. So mycorrhizal symbiosis are now recognized as play a fundamental role in providing the ecosystem services due to these beneficial functions like plant fitness, then resilience against environmental stress, then nutrient cycling, as well as soil quality. So all these are the different parameters the mycorrhiza can influence in an ecosystem. So coming to this action against pathogen, so whether the mycorrhizal plants give any protection to this against any diseases, definitely the mycorrhizal plants are known to give protection against plant diseases, particularly soil-borne diseases. Like where these phytohormones like salicylic acid and jasminic acid plays a very important role in inducing systemic resistance during pathogen invasion, and mycorrhiza have shown to enhance the level of these hormones significantly when compared to non-mycorrhizal plants. Okay? And this mycorrhizal fungi also played a very important role towards improving uptake of nutrients and improved resistance to plant against soil-borne pathogens. So this is the integrated response of AMF colonized plant against pathogens. So the process involved is activation of defense response through this localization of induced resistance, systemic resistance, through this priming of genes like jasminic acid and salicylic acid, then cell wall defense, then defense-related protein production, then defense-related enzyme activity, enhanced production of phenolic compounds and benzoazenoids actually. And in addition to that, the mycorrhiza also directly compete with the resources, particularly competition for photosynthate, then root space, then infection site and nutrients. So if mycorrhiza is early in colonizing the host plant, then it avoids the entry of pathogen actually. So that is how through direct competition, the mycorrhizal plants can inhibit the entry of pathogen actually. And altered rhizospheric interaction is also another one important aspects where the change in root exudate qualities and composition which modulate hormonal imbalance, then influence rhizospheric microbiome, promote PGPR population. So all these are brought about by mycorrhizal plants when compared to non-mycorrhizal plant. Then altered root morphology, and because the mycorrhizal association is known to enhance lateral branching and damage compensation is very fast when compared to non-mycorrhizal plant. So that is how it gives bit a edge over non-mycorrhizal plant. Then enhanced nutrient acquisition, of course, greater plant growth and increased tolerance to pathogens is through this what you can say, the nutrient which if the plant is healthy, definitely it can have the ability to resist the entry of pathogen into the host plant. So that is how there is an integrated approach within a mycorrhizal plant and a non-mycorrhizal plant. So we have conducted an experiment wherein we want to see how the mycorrhizal plant can influence the formation of diseases within, or how the plant growth will be there when the dual inoculation is done. We have taken the Glomus aggregatum as one of the inoculum, mycorrhizal inoculum, and Fusarium woodum, which causes wilt disease in pigeon pea, that is Kajanus kajan. And we saw that the dual inoculation enhanced the growth of the plant, and only the plants which are inoculated with the Fusarium showed the disease, and the plant which are only single inoculation didn't show that much this one growth. Actually, dual inoculation showed a good growth actually. And we also wanted to see the effect of mycorrhizal association in managing black bundle disease caused by Cephalosporin acremonium. You can see here, we found that so the disease, the mycorrhizal, the pot which were inoculated with the Glomus mossae and Cephalosporin acremonium showed only 16% of disease when compared to 60% of disease when we inoculated with as a negative control. So both this Glomus mossae as well as this Lewis, Aclosporium Lewis showed 16% disease only. That is, you must see, you can say that it has shown a good effect in suppressing the plant disease. Coming to this plant growth promotion by AMF, definitely the AMF considered to be the transporters of various elements from the rhizosporic soil and to the host, whereby it increases the surface area of the nutrient absorption, and it interact and accommodate many of other soil microorganisms through their hyphae. Thus, the term rhizosphere is considered to be as a mycorrhizosphere actually. Okay? And we wanted to see, is there any effect of this biomass enhancement in case of Piper longum. So we used Acolospora fovata, then Glomus fasciculatum and Gagaspora margarita as inoculums, and we found that. So when compared to control, you can see the root number of leaves produced varied, and root length also varied. You can see there is no significant difference between the control as well as this one, and but in the shoot length, you can see there is not much, but in dry, the whole weight of the plant, if you consider, so the growth enhancement was on par with the control actually. Okay, and coming to this efficacy of this growth of on the Centella asiatica, which is the medicinal plant. So we observed that the drastic, almost Gigaspora margarita has enhanced the number of leaves in drastically when compared to control. You can see here, and shoot length also increased when compared to control. So the growth as on a whole, actually, when we recorded all three plants, all three mycorrhiza, there is Glomus margarita, then Glomus fasciculatum, and Acolospora fovata. So all these three are known to be a good inoculum for increasing the growth of Centella asiatica. And another one, we conducted an experiment to see the efficacy and infectivity of four native Glomus fungi, particularly Glomus fasciculatum, Glomus mossae, then Gigaspora margarita, and Acolospora levis. You can see this is the control. When compared to control, all the four inoculated plants have shown very good growth actually. So if you see the dry weight, in control it is on shoot and root dry weight. Totally it is 2.6, and all other dry weights were drastically increased actually. You can see here, and height of the plant also showed a drastic increase, and the pot inoculated with this Gigaspora margarita and Acolospora levis showed a good growth. when compared to other plants. And we wanted to see the growth and nutritional factors of Vigna radiata by inoculating Glomus fasciculata and Acolospora leaves. We found that the fresh weight and dry weight, this is sterilized control and unsterilized control, these two. And this sterilized Glomus fasciculatum and unsterilized Glomus fasciculatum, then sterilized Acolospora leaves and unsterilized Acolospora leaves. You can see here in unsterilized and this sterilized, there is a drastic difference when in inoculated plants when compared to control actually. And even in the nutritional status, we wanted to see this shoot nitrogen and phosphorus. So there is huge increase in case of Acolospora leaves actually when compared to control. So these experiments can help us in selecting the suitable inoculum for a specific crop actually. So in this case, we found that this Acolospora leaves is considered to be one of the important inoculum, both which increase the root weight and shoot weight as well as the nutrient status of the plant actually. Okay? So the response of plants to essential and non-essential elements in terms of their threshold level. So at the optimum level, these are the essential. If the essential elements are deficient, and if the essential level are toxic to a plant actually. So this is at the low level, then this is at the high level. High level also it is toxic. In low level also it is what you can say toxic. But the non-essential elements, you can see at the low level they are very good, and at high level they have become toxic actually. So the mycorrhiza are also considered to be one of the important components where heavy metals, to elevate heavy metals actually. So whatever the heavy metals which are there in the soil, if the plant is forming an association with mycorrhiza, the mycorrhizal spores and mycelium is known to accumulate actually those heavy metals within their mycelium, and they will not transfer these heavy metals to the root actually. And in the absence of these mycorrhizae, these heavy metals can directly enter in the plant, and they can cause so many diseases. If it is accumulative in nature, that in turn will accumulate if the plant is edible actually, and biomagnification takes place, and that may lead to various health hazards in humans actually. And even if the plant is known to have a deleterious effect with these heavy metals, and the plant growth will be retarded actually. So this mycorrhiza plays a very important role in accumulating these heavy metals within the mycelium and the spores it produces actually. So this is how they help actually. So we also wanted to see this the effect of fly ash, because fly ash is considered to be, as not now actually, earlier it was considered to be one of the important component where the coal burning for this generation of thermal power plant near Raichur, we wanted to see whether it has got any effect. So we conducted an experiment by amending this fly ash in different concentration, that is 10%, 20%, 30%. And we used the Glomus aggregatum, whether it has got any effect. We found that the plant, the fly ash amended plant with Glomus aggregatum proved to be very positive response when compared to non-inoculated plants. Actually, we tried with this Caesar ranatinum, that is chickpea, and Cajanus cajana, that is pigeon pea. So coming to this mycorrhizal inoculum production, as of now, I think trap culture is one of the method where the pure culture can be obtained. You can isolate this native. If you want to go for mass cultivation of this mass culture of native soils, you can isolate the spores, and you can use a trap crop of any interest actually. Either you can use sorghum, or you can use ಇನ್ನೊಂದು 5 ನಿಮಿಷ. You can use sorghum or any monocot plants for mass multiplication, and the whole root along with the rhizosporic soils, you can use that as a inoculum actually. So coming to this future perspective, what are the future aspects one can look? So arbuscular mycorrhizal fungi can be used in sustainable agriculture, then plant growth and productivity, then you can use for increased mineral uptake of new mineral nutrients, water uptake, managing rate of photosynthesis, accumulation of secondary metabolites and active ingredients. And also abiotic environmental stress tolerance. Then you can look for disease resistance, soil health and fertility, ecosystem functioning and biodiversity, then reclamation of degraded land and bioremediation, prevailing soil erosion with the help of this because they are known to produce glomalin, then mitigating global warming and climate change, because whatever the association is mainly for nutrient acquisition, and they also compete for carbon source. So whatever the carbon source is fixed in the plant, the same will be transferred to the mycorrhiza. So likewise, they are known to mitigate the global warming. That is the main aim. And in conclusion, I can say that arbuscular mycorrhiza can be used as a biofertilizer to enhance ecosystem functioning than agricultural productivity without disturbing the ecosystem balance or ecosystem environment and ecosystem. Then arbuscular mycorrhiza characteristics are make them potential tool to be used for sustainable management of agriculture and environment. Okay. Therefore, the field inoculation of AMF, depending upon the type of plant species and their environmental conditions, could be an effective alternative for agrochemicals. Okay. So these are the first. Thank you. So